started his, here we go. Scott started his academic journey at the Johns Hopkins University, getting his BA there in biochemistry. He then stayed at Hopkins to get an MD PhD in pharmacology. And then after that, he ended up coming to the NIMH here and has been here for a number of years, starting his lab and really focusing on studying the neuropeptides oxytocin and vasopressin. And since 1996, he's been the principal investigator and chief of the section on neural gene expression in the National Institutes of Mental Health. He has a list of awards that I won't spend a lot of time talking about. I think the best one to highlight here is he was given the Outstanding Mentor Award in both 2010 and 2011. And I think that you can see that he really engenders a lot of loyalty in the people that have worked with him, because he has several of his trainees spanning back 20 years that have come to listen to this talk. And he did promise to give us a short sort of story of his academic career, as this is his last talk as an NIMH section chief, because he will be leaving the NIMH next month. So with that, Scott, if you want to take it away.
Well, thank you very much, Sarah. It was a pleasure to have you as a fellow in the lab and to still work with you as you're working in the core facility with Ted. Let's see if I can get this going. Okay, is that okay with everybody?
Looks good.
Okay, so thank you again for the very kind introduction, and I'm glad that many friends have showed up for, I guess, my last talk. The title I gave was Hippocampal CA2 Area is Essential for Social Memories and Aggression, and I would add subtitle, or What a Great Trip It's Been, and I'll hope to show some of that briefly in the beginning and show how that might relate to how things have turned out. So I had the fortune of being a graduate student in the Division of Psychopharmacology of the Department of Pharmacology and Experimental Therapeutics at the Johns Hopkins School of Medicine in the late 70s. I was mentored by Michael Kuhar, and we came up with this approach to look at in vitro the localization of receptors by autoradiography. And basically all we did was took a mildly perfused animal, cut tissue sections, put them on a slide, incubated the sections in a solution containing a radio-labeled, at that time tritium labeled ligand, plus or minus the dispenser to get for the controls. Then we, after various incubation times, we'd rinse, then blow dry. And we found that by scraping off the tissues and counting them, we could show that this process had not harmed the receptors themselves. We could do nice kinetic studies on the tissues, so that validated the fact that we were actually looking at receptors on tissue sections. Most people have been doing them in homogeneous to that point. And then we could take those other slides that we didn't scrape the sections off, oppose a motion-coated cover slip to a slide, let that expose for weeks or months, and then develop the emulsion. And we could get images like these opiate receptors in the rat brain using dihydromorphine, or benzodiazepine receptors in the human cerebellum. This center one is the actual autoradiogram. This is a Nissl stain. And then this is control that had excess benzodiazepine to block the treated benzodiazepine. So that was a lot of fun. That division was headed up by Saul Snyder, and it had Mike Kuhar, Joe Coyle, Elliot Richardson. So it was really a lot of fun. I was lucky to be there. And so I did a lot of things with in vitro receptor autoradiography because it was a new technique, and there were lots of drugs that were labeled coming out there, so we could map anything we wanted. So I looked at receptor localizations for diffusible drugs, for receptors for neuropeptides and other transmitters such as GABA and norepinephrine, receptors for various receptor subtypes such as delta and mu opioids or various benzodiazepine receptors. And we looked at receptors in human tissues and other primate tissues, and we could even demonstrate that these receptors underwent axonal transport. I would be obviously remiss in saying that other great things happened while I was in that division, including the fact that I met my future wife. She was working down the hall in Joe Coyle's lab. This is Michael Kuhar at our wedding, and this is Michael Brownstein, whose lab I spent a summer in as a medical student and would actually come back to NIH after doing my medical internship and neurology residency to work in his lab. So this lab allowed me to continue doing neuroanatomical studies and using some somewhat similar approach we started doing in situ hybridization, which was relatively new, believe it or not, at that time. We used first radiolabeled oligonucleotides and then eventually RNA probes, both radiolabeled and colorimetrically labeled. We started off looking at things like corticotropin releasing factor, vasopressin, and oxytocin in normal mice and rats, and in ones that had been manipulated physiologically by, for example, stressing or giving them lots of water to drink. With Stafford Lightman, who came on sabbatical a couple times from England, we looked at the effects of stress and other manipulations on CRF, oxytocin, vasopressin, and so forth. Even had a chance to work with Peter Seberg, and this was also done with Tom Zeller, who was also in the lab of cell biology, looking at expression of the GnRH gene in mice and in the mutant mouse. So a little background about oxytocin and vasopressin. They are 9 amino acid cyclic neuropeptides. In mammals, they are expressed in the supraoptic or SON nucleus and in the paraventricular or PVN in the hypothalamus. Vasopressin is also made in the clock nucleus, the suprachiasmatic nucleus. And in some species, oxytocin is expressed in the medial amygdala, for example. More refined and sensitive in situ methods have shown that, for example, you can find oxytocin cells scattered here and there, even in the cortex, but at really low levels of expression. So these these neuropeptides act in the CNS through 3 receptors: the vasopressin 1A receptor or V1A receptor, the vasopressin 1B or V1B receptor, and the oxytocin receptor. Vasopressin was identified over a century ago through its release from the pituitary, the posterior pituitary, and effects on salt and water balance. Similarly, oxytocin was discovered back then through its effects on milk letdown and parturition. Other effects have been found as well, and including and importantly, its effect on ACTH expression and release from the anterior pituitary, which I'll talk about extensively coming up. Over time, these hormones have been associated with various behaviors such as stress responses, sexual behavior, paternal and maternal behaviors, and so forth. We became interested in social recognition and social aggressions, territorial and maternal. Oops. Well, the vasopressin 1B receptor has a long history actually in the laboratory of cell biology. The initial discovery of the receptor was found in this lab in 1984 by Ferry Antony, Steve Lollet, Anne-Marie O'Carroll, and others in Mike's lab, cloned the rat V1B receptor gene, eventually the mouse one as well. Scott Werstinger studied the behavior of the knockout mouse that we made and showed a decrease in aggression as well as social recognition in this mouse. Steve Lollet went on to study with his wife, Anne Marie O'Carroll, went on to study extensively the effect of the knockout on the pituitary adrenal axis response to stress. And even more recently, in 2017, members of Ava Mezzi's lab, who used to be in the laboratory of cell biology, but now has had her own lab in the Dental Institute, showed that vasopressin can stimulate the proliferation and differentiation of red blood cell precursors even before erythropoietin can do its thing. So anyhow, we made this knockout for the V1b receptor, and Scott looked at this. He brought behavior to the lab and showed that compared to the wild type in red here, the knockouts did not respond with aggression to putting a strange male into their home cage, at least not very much. And even after 3 attempts with this strange intruder, they didn't attack not as many attacked, and those that did had a longer latency to attack with reduced associated attack behaviors. He also showed that after a 30-minute separation from having first seen a female mouse, the male mouse could remember in the wild types and hets that he'd seen this mouse before, because he spent less time sniffing the familiar as opposed to the novel, but the knockout prevented this memory. Interestingly, defensive aggression was not affected by the knockout, but a retaliatory aggression was affected. Also, and that was done also with Heather Caldwell, who was in the lab, and they also looked at maternal behavior, and that was also, this maternal aggression was also reduced in the knockouts as compared to the wild types or the hets. Predation for a cricket was normal in these mice. All attacked the cricket, and they learned that the cricket that they'd never been exposed to, they learned to, I guess, like the taste. And so after 3 tests, they were even attacking more quickly than originally. So where was this happening? Well, in 2006, we found that the V1b receptor is actually expressed in a small region of the hippocampus. Back then, we used radio-labeled compounds. This is an updated version using 3 fluorescently labeled probes. This is work done by Jun Song a little while ago. And we use a marker for CA1 in the hippocampus in these pyramidal cells, and BAC1 for the CA3. You can see this gap between them, and that's CA2. This was first described by Laurent Deneau in 1934. And you can see that the vasopressin 1b receptor is expressed in this gap. There's a pretty nice cutoff between CA1 and CA2 for almost all markers that you look at, whereas there's a substantial overlap between CA3 and CA2 for some reason. So at that time in 2006, we proposed that perhaps this particular hippocampal field helps to either form or retrieve memories of social encounters, much as other hippocampal pyramidal cells establish memories of place. Our results with the V1b knockout mouse could be explained if each encounter failed to associate context from the social experience. Just as a side note, this part of the hippocampus has been associated with various human diseases, especially schizophrenia, where there are deficits in some of the inner neurons as well as volume of that region. And before I go on, I should also mention that making these early knockouts, we made an oxytocin knockout and the V1b knockout, wouldn't have been possible without the expert technical skills of Emily Shepard, my long-term technician who joined me only a year or 2 after I joined the laboratory of cell biology. Also, so as you recall, social recognition is impaired in the V1b knockouts. Heather Caldwell started work in a lab and finished it when she went to Kent University, showing that NMDA lesions would abolish the social memory. These are specific CA2 lesions compared to the sham injections. And this was confirmed by the Siegelbaum group, Pitti and Siegelbaum in 2014, where they used the tetanus neurotoxin light chain that they expressed to the CA2. And they also showed that inhibiting firing there inhibited time spent sniffing. So what about aggression? So this is a work that was led by Barney Pagani in the lab. And he took knockout mice that don't express V1b receptor. Here you can see the wild type expression in the CA2. They don't express, the knockouts don't express. We made a lentivirus that expresses the V1b receptor, and he injected them into 3 sites bilaterally, 6 sites altogether. You can see a blow up of that image showing expression now in the knockouts. And as opposed to knockout, which did not show much in the way of aggression, we could restore much of the aggression after replacing the virus into the CA2. Now, we didn't obviously cover the whole CA2 from front to back, but enough of the area to return aggression. Sarah Williams, who was in the lab, as you heard earlier, was able to take a inhibitory dread, inject that into the CA2, give them CNO, and was able to inhibit maternal aggression, shown by increased attack latencies, decreased numbers of attacks, and time engaging in aggression. So how does this, how does the CA2 get involved in aggression? Leroy and Siegelbaum's lab did a nice series of studies. I'm not going to go through all the data or I'm not going to go through all everything on this slide, but basically it came down to showing that the CA2 projects to the dorsolateral septum, just as you saw in Sarah's picture here, which then inhibited cell firing in the ventrolateral septum, which inhibited firing in the ventromedial hypothalamus. And all that leads to an increase in aggression. So if you eliminate the input to the dorsolateral septum, you get the opposite, a decrease in aggression. One of my favorite series of experiments was initiated by Adam Smith, Sarah Williams, and Adi Sarnablat-Saba. What they did was they injected a virus into the PVN that would express channel rhodopsin that would then be carried from fibers of the PVN to the hippocampus. Then we could put a optic fiber into the CA, just above the CA2, and start shining a blue light on that. And then we would do that during different aspects of behavior and see what that did to the animal's behavior. So the first paradigm we used was to have the subject male mouse sniff a female mouse for about 5 minutes, and then remove that female for 2 hours, and then either put back the familiar or the novel mouse. We knew that they could remember the familiar mouse at 30 minutes, and we also then found that they could not remember them after 2 hours. That's shown here by the sniffing duration and by this ratio. If we stimulated the mouse during the initial exposure and then on replacement of a mouse in the cage, it could then remember the original familiar mouse. This did not work for an object. So object memory was not influenced by the stimulation, nor was there any change in the time spent sniffing the novel mouse as well. So when did this happen? So in the first experiment, we stimulated both during acquisition and recall. So when we did it only during acquisition, we could see a decrease in sniffing of the familiar, shown here, indicating a recall or memory of the original mouse. When we did it during the recall phase and not during the acquisition, then there was no effect. So it's important to do it at least during the acquisition. It might have helped to do it also during consolidation, but we didn't test that. So those were 2-hour, 30 minutes and 2-hour exposures. What happens if we did longer exposures? So we knew that we'd already shown that after 2 hours, they could remember after being stimulated, not if they weren't stimulated. At 24 hours, they still could remember the familiar. And even at 7 days, they could remember the familiar mouse. We didn't go any longer. I doubt it was permanent, but it would be interesting to know how long that memory does persist. An interesting experiment they decided to do was something called retrograde interference. And you can see how this test would be important for any society. So what happens is normally in the paradigm, the subject mouse is presented with a stimulus. The stimulus mouse is removed after 5 minutes for 30 minutes, then replaced, and it will remember that mouse. But if after 5 minutes you put in a novel mouse for 5 minutes, then remove it, and then 20 minutes later test the original familiar mouse, it doesn't remember the original familiar stimulus mouse. Whereas if you stimulate the subject during the initial exposure, it will now remember through the retrograde interference the presence of the familiar mouse. And like I said, this would be important for society. You wouldn't want to forget the person you just talked to if somebody else walked up to you. We could also show that this was specific for the V1B receptor. We used a 2-hour paradigm, and after we infused 10 nanograms of the V1B antagonist 5 minutes prior to the stimulus, we would block the effect of the stimulation. So this Vasopressin PVN to CA2 stimulation enhances memory of social but not inanimate objects by action during acquisition, not recall, by over 80-fold following one trial, by reducing social retrograde interference caused by intertrial presentation of a second female mouse, and that is blocked by a specific pharmacological antagonism. So I was at a concert at the Strathmore, and after we'd been doing some of this, and I thought, could we actually induce partner preference in these laboratory mice, not known to have any sort of monogamy or partner preference? Could we do this by direct stimulation of the PVN to CA2 vasopressin projections during mating? So I brought this back to the lab, and the team came up with this approach, which was to retrogradely transport a dread, an excitatory dread from the PVN, from the CA2 back to the PVN, and then administer CNO directly into the PVN prior to the overnight cohabitation of the male with the female. And what they found was that in those animals that were given the GQ plus CNO, they preferred the next day increased time of contact with the partner as opposed to the stranger. As you can see, there's no difference in the mice that were given the control virus, even though they got CNO. There was a much greater increase in allogrooming in the to the partner as compared to the stranger. And there was an increase in side-by-side contact with the partner as opposed to the stranger. This is quite reminiscent of what's seen in the monogamous prairie voles. So to me, it says that that pathway is still intact. It's probably not being excited or used, at least not in the laboratory settings. So another of my very talented fellows was Jin Lee. He created a knock-in of the Cre recombinase gene into the vasopressin 1b, in front of the vasopressin 1b promoter, so that the V1b promoter would drive expression of Cre in V1b cells. And so you can see in this upper right-hand panel that they're both, in this double in situ, there are both messages for Cre as well as V1b in this CA2 area. And this is just a higher level showing intermixing of the two transcripts. So one of the first things we did, this is something that Sarah did, was to take a line of mice in which the R1 subunit of the NMDA receptor is floxed and cross that with our V1b Cre line. Normally in the wild type, you see this prominent expression of the subunit in the hippocampus, but after crossing with the CA2 expressing Cre line, you can see we lost much of the expression there. And obviously we felt that this was going to lead to a decrease in aggression and a decrease in social behavior. But to our surprise, the elimination of this subunit in the CA2 did not affect social recognition. Behavior was the same, either in the hets or the knockouts. Social novelty was also not affected. So this is a chronic adjustment, probably. We don't know actually if we had done this acutely, what would have happened to the behaviors. Adi decided that she would look at cell firing patterns in the CA2 using the calcium sensor GCamp6S. So this was injected into the CA2 of mice, and then a mini microscope could be attached to look at cell firing under UV light of the CA2 neurons during behavior. You would see these individual cells shown up here in the upper left, as well as these calcium currents or calcium responses in those cells. And you should, as I said, follow what was going on during contact with another mouse or with an inanimate object. So one of the behavioral tests we did was, or that she did with a bunch of talented postbacks, was to look at the responses during repetitive encounters with a mouse number 1 through the first 4. And then on the next trial, a novel mouse was put in, and then 45 minutes to 60 minutes later, the original put in. And you could do the same thing with 2 different types of inanimate objects as well. So there's only a few mice, like 3 or 4 mice in the study, so maybe the behavior is not terribly pronounced in terms of sniffing, but you can see there's an increase in sniffing after the novel mouse is put in. And you can see that this is paralleled by a decrease in the number of active neurons in the CA2 with time as familiarization occurs, and then there's a bump with the novel mouse put in. Same with the event rate. When we look at the object habituation, dishabituation, really not much going on, maybe a slight decrease or a decrease in the second object exposure, but nothing's really happening. Nothing happens with the novel presentation, novel object presentation. Same with the event rate. So she concludes that the CA2 neuronal activity recapitulates the sniffing behavior by the experimental mouse, so the conspecific, but not of the inanimate object. That short-term social memory is encoded by a mechanism of repetition suppression and sparsity, creating sharper, efficient representation of the social cue. And that different coding schemes serve this dorsal CA2 and its role as a hub in the hippocampal microcircuit, integrating multiple inputs and sending them downstream appropriately. So over the past year, she's been finishing up some of these studies and has actually recently just completed the technical work, and now she is evaluating the results that she's had. And one of the questions is, how are neuronal responses changed in the knockout mouse, the V1b knockout mouse? And the initial results show that there is no change. I mean, they do not show a change. They don't show a habituation as opposed to the wild types, which did show the habituation, and there's no bump or increase when the novel is put in. And this goes for both the number of active neurons. But for the, I guess for the average event rate, there is actually a decrease in events occurring with no bump, even a decrease in the when the novel is put in there. So very different from what you see in the heterozygotes. And she's analyzing data to see what more can we learn by employing, or she did employ more complex behavioral paradigms such as alternating mice and or inanimate objects. So it turns out that oxytocin receptor and the vasopressin 1b receptors are co-localized in CA2 pyramidal neurons. And here you can see in the top row, lower power, and in the bottom row, higher power magnification, showing vasopressin 1b expression in the CA2, oxytocin expression in the CA2, but as I mentioned before, well, I guess I didn't mention, but oxytocin also, the receptor also extends into CA3. The oxytocin receptor is much more widely distributed in the brain than the V1b receptor. And in simultaneously looking at these, you can see they're overlap in the CA2, but not in the CA3 where the oxytocin receptor extends. So we had the question as to what would happen if we knocked out the oxytocin receptor only in the CA2, and we could do that using our V1b Cree line. But first we needed a mouse line that had the OTR floxed, and again, Jin Lee had made this mouse that actually it's become very popular in many labs. And we, in the presence of Cree, these floxed introns are removed. And part of the reason we decided to look at the local inactivation of OTR in the hippocampus was because other people had been using this mouse and using viruses to knock out the OTR in this area. But it's hard to confine the virus just to the CA2. And OTR is also made in interneurons as well. So it wouldn't be as clean as just knocking it out of the pyramidal neurons. So this is what happens. So this is the expression of V1b in the wild type, and this is OTR in the wild type, both in the CA2, but OTR into the CA3. If we have this sort of a het or just oxytocin knockout in which V1b is just heterozygote for Cree, but the OTR is floxed, homozygous, so you can see that the one Cree allele knocks out the two floxed alleles, so we no longer have oxytocin in this OKO. Whereas if we have a double allele for Cree and double flox for OTR, we now have no V1b because the Cree takes the place of V1b, and we have very sparse OTR left, perhaps from the interneurons or from some of these CA3 neurons that invade CA2. So one of the experiments that Adi and others performed was to do the 30-minute intertrial interval between familiarization with a female mouse, and then a third exposure to differentiate between the novel and the original mouse. And not surprisingly, the OTR knockouts did not show recognition between T1 and T2. but really surprisingly, the double knockout did. And it was able to remember the original stimulus. Neither line had any difficulty distinguishing between the novel and the social in the T3. So what was going on? So I should step back a little bit and mention that early on, maybe 15 years ago, I was alerted to the very interesting work that Serena Dudak in her lab was doing in the CA2, looking at LTP and the difficulty in inducing LTP in the CA2, and we started a fruitful collaboration and a nice friendship. And as you can see, when they took sections from mice and looked at the ability of either oxytocin analog or vasopressin 1B analog to facilitate or enhance Schaefer collateral stimulation of CA2 neurons, that these agonists could do so in the CA2, but not where the receptor is not in the CA1. Same with oxytocin analog, no stimulation or no enhancement in CA1, but there was enhancement in CA2 and also in CA3, which is also where this receptor is expressed. So her lab also showed that if you took the knockouts, they did not respond to this, to the agonist for V1B, but did still respond to the agonist for oxytocin, whereas the OTR knockouts did not respond to the OT agonist, or oxytocin agonist, but did respond in wild types or to the V1B knockouts. So what was going on? So Kai Zhang Duan in the lab showed that with Schaefer collateral stimulation, that there seemed to be a chronic compensation for the chronic loss of oxytocin and V1B, so that there was actually an increased effect of the Schaefer collateral stimulation on CA2 excitability, and it seemed to be presynaptic based on this paired pulse ratio. And there was no effect on fibers going from CA2 to CA1. So we don't know what this compensation is. It would be nice to do, say, a gene-seq, RNA-seq or something on the CA2 in these situations, or CA3, but we haven't done that. We did want to see if acute inactivation using drugs had the same effect, and no, it didn't. All three situations, the V1B antagonist, the oxytocin, and the combined antagonist decreased the amplitude after stimulation or during stimulation. Another experiment that we've just finished up, and I don't have all the results for yet, is our investigation of the fasciola cinereum, which was first described in 1882. Turns out that the CA2 is not the only region in the hippocampus that expresses V1B or other CA2 markers. Another place that does that, or the only other place that does that, is the FC, fasciola cinereum. Actually, if you look at a brain that a brain anteriorly, you can see that there's a sort of a continuation between FC and CA2, and then as you move more posteriorly, the CA1 protrudes and pushes the two apart. So this is where the CA1 has pushed them apart, and you can see expression of V1B as well as the marker amigo2 there. Here's a CA1 marker, and you can see the CA2 marker, V1B here and here in CA2. So what we did was cross the V1B Cree line with a line that makes GFP analog, and that's retained in the nucleus. You can see expression of this GFP in the dorsal CA2 as well as in the FC. So what we did was we crossed these lines, and Sue Hyun-Lee punched out these regions, FC and the dorsal CA2, lysed the tissues to isolate nuclei, did FACS sorting, then isolated the RNA, made cDNA libraries with barcodes, excuse me, and then we sent them to the NIH sequencing center for sequencing of up to around 100,000 base pairs per cell. And then we looked at these bioinformatically, and you can see that there's a very similar pattern using these UMAPs of the dorsal CA2 compared to the FC, but there are differences. You can see that the FC has this different pattern here. And then the bioinformatics indicated that there are 7 cell types, and here they are shown by color. So we don't know what all these what these cell types do. We're still analyzing and doing gene ontology studies or investigations. But this top row shows some examples of cells that express more or have more cells expressing either one or both in the FC, and the bottom row shows genes that are expressed more in cells in the CA2 than in the FC. So one of the cells that's expressed more in the FC than CA2 is the gene for substance P. You can see that it's expressed more in these FC types, type 7, 4, and 5 compared to the other types. And here's an occasional cell in the FC that's expressing TAC1. We've been looking at these by high-plex, and we're doing up to 24 genes on top of each other, see if we can see any patterns there. Here's one of the specific type 7 genes, platelet-derived growth factor receptor alpha. Again, it has a couple cells here and there in the FC. We don't know what the roles of these genes there are yet. One gene that interested us was the sodium voltage-gated channel alpha subunit 1, or SCN1A. And in the FC, the expression levels are lower than in the dorsal CA2. So Kaisong looked at the electrophysiology of these cells and tissue slices. Here are, we crossed our B1B line, B1B Cree line with TD tomato line, and so we can see them in culture. And here you can see the FC or the CA2 neurons, and at a given stimulus, the FC cells hardly ever fired as compared to the CA2 neurons. And the FC tended to have a resting membrane potential more negative than the CA2 neurons. Again, we don't know what the significance of the different firing patterns are at this point. But we did do a little behavioral study in which we injected the DREAD GI into the FC and then administered CNO, and we measured social recognition, and we did not find an effect on social recognition either by sniffing duration compared to the wild types or wild types with CNO. Although we did notice and replicated the fact that in the presence of CNO, these male mice showed a much increased amount of mounting behavior. So again, we don't know why, but that's an observation we made from this. This is not something that we have seen in any of our stimulations of CA2. Curious thing about the FC is it is absent in a mouse strain, the BTVR mouse, that is used to study aspects of the autism spectrum disorder. So last but not least, in the future, we hope to come back with some data. This is a study that was initiated when Jin Lee was back in my lab on sabbatical, and he made CRISPR constructs to insert Cre recombinase into the peribols of the vasopressin, oxytocin, and oxytocin receptor genes. And then Adam Smith and Dev Manoli are now pursuing this. They now have lines of the OTR gene with the Cre knock-in, and they're studying various aspects of the OTR expressing cells and their projections to say the commons in other areas, and using dreads and other things to be expressed in only the OTR neurons. So that will be things coming out in the future. If you want to see things that are happening now as well as in the future, I would also recommend these really excellent CA2 investigators that are picking up on some of the things that we've got started. And finally, I'd just like to thank everybody who's made my career at NIMH possible. The final lab configuration consists of the excellent staff scientist Adi Sayar-Butsabi and the excellent fellows Su-Hyun Lee and Kai-Zeng Duan, both of whom have jobs outside of NIH in industry. Adi will be in Dr. Mara's lab. Emily Shepard, my longtime excellent biologist, Jun Song, who joined the lab over a decade ago and has been really good at doing our hybridization histochemistry and other technical assistance. And then the final two postbacks, Oliver Holmes and Savannah Aguilan, I think I spelled her name wrong, who I gotta say, I feel sorry for, because they joined us when the lab was undergoing the COVID restrictions, and now the lab is closing, but they've been a great help during that time. So I appreciate that a lot. Of course, previous fellows, many of whom I mentioned, recent excellent collaborators. And of course, none of this could have been done without the great NIMH Transgenic Core Facility, led by Jim Pickle and the Animal Research Program under Dr. Raber with the assistance of Julia Drake. So with that, I'll stop. And if anybody has any questions, I'll be happy to try to answer them.